Well, good evening, everybody. It's certainly a privilege and an honor to be able to share with you the wonders and mysteries of God's living creation, especially when it comes to the biological systems. I'm a zoologist, as Ken said. Now, what's a zoologist class? It's just us here this evening. What's a zoologist? Zoologist is somebody who studies zoos, okay? And I find that's very boring, okay? So as Ken said, I'm a zoologist. I study animals, and the animals I study are parasites. How boring is that? And so I don't usually talk about parasites, because especially if you're going to have supper, especially if you're going to have spaghetti. But I found out early on that you don't have to go to France to study parasites. Thank you. Thank you. Okay, good. This termite goes into a bar and says, "Hey, is the bartender here?" Termite eats wood. Is the bartender here? It's what we call a time release joke. Some of you wake up about 2 a.m. Got it. So anyway, I'm standing here this evening as one who has been spoon-fed the theory of evolution in my undergraduate days in Colorado, and then as I went to graduate school, the University of Northern Colorado, I was taught evolution as a fact. My late professor, my major professor, Dr. Gerald Schmidt, said, if you want to be my graduate student, he said, you must take a course in evolution. Well, by that time, my faith was pretty strong in regard to creation versus evolution. So I said that would be fine. And so I took an evolution class. It was an upper level class, graduate level class. And for an entire semester, what did we talk about? If there's no facts of macroevolution, that is vertical evolution, then what did we talk about? And the answer is we talked a lot about genetics. Well, I love genetics, and genetics is hardly any issue in creation evolution. Genetics simply shows how God has produced and created the varieties of animals and plants that we see today. It has nothing to do with macroevolution, and so really this evolution class was advanced genetics, and I enjoyed it very much, and I actually got a good grade too. So this evening, just a brief time that we have, we're going to talk about the brain's amazing ability of visual perception. I taught anatomy and physiology at Pensacola Christian College for a number of years, teaching pre-med anatomy, biology, cell biology, parasitology, medical microbiology, all of those really boring classes, but it gave me an appreciation for God's living creation. And so what we're going to look at is just this small area of visual perception and just see that it really is quite a compelling evidence for creation. So let's go ahead and do that right now and start with what I call Vision 101. Now, all of us have had general biology in high school. Remember that? Set the Wayback Machine way back when you took general biology in high school. We all had to have it. And so a lot of this, what you'll see is just very general overview, and it gets into a little bit of detail, but we have to set the stage before we talk about the subject tonight. So this is the electromagnetic spectrum. This is how the Creator has designed us as vertebrates, particularly people, to see the light. What is the light spectrum there? Obviously, we cannot see in the FM and AM range, nor can we see the gamma rays, and I'm thankful for that. If we saw all of the electromagnetic spectrum, we would just go crazy. And so the Creator in His infinite wisdom has designed us to just see, as you see right there, the visible spectrum, anywhere from 397 to 723 nanometers. And so that's the units of measurement there that include the visible light spectrum. Now, neurons are a nerve cell. The 3-pound brain that's holding our ears apart are composed mainly of neurons, but there's also special cells called astrocytes, the protoplasmic and fibrous astrocytes, which support the neurons here. And when I was in graduate school, I did a paper on these astrocytes. They're really quite amazing. And unfortunately, sadly, when people get brain cancer, it's usually those astrocytes that cause the problem and become cancerous. The nerve cells that you see there do not generally become cancerous. Well, this is a cross-section of the eyeball. We've seen this 100 times, and this gives you just a basic overview of the anatomical features of the eyeball. It really is a wonder of creation when we look at it. And what we're interested in as we're looking at the eyeball is right here, the retina. The retina is very, very important. It's the sheet of tissue that covers the backside, basically, of the eyeball. And then, of course, we have the optic nerve here, optic nerve about the diameter of a pencil, and we'll get into that just a little bit. But this is the eyeball with the lens and the iris and so forth, and the all-important retina, as I say, that we're going to look at, no pun intended, right now. And so it's not asking the question, how thick is the retina, but really how thin is the retina? How thin is that photosensitive sheet of tissue, living tissue, that's basically a computer, but it's a lot more, it's a supercomputer. How thin is it that is in the back of your eyeball there? Well, what we're looking at here is a dime. And if you cut this dime in half, lengthwise like this, in half, then a dime is 1 millimeter, so cut it in half and you have what, class? A half. See how easy science is? You have a 0.5 millimeter, okay? Now again, here's a cross-section of the eyeball. Here's the retina that we're talking about. How thin is the retina? It's only about a 0.5 millimeter in thinness. That really is quite amazing because when you look at the retina, you find all sorts of layers of cells in that 1/2 millimeter. All sorts of layers of cells. And I think about 20 years ago, they found yet another layer of cells that make up the retina. So it really is incredible, this very thin sheet of tissue that is more powerful than any kind of supercomputer and all of the functional cells that it has. So the eyes convert energy, that is photon. Who thought of the word photon? Sir Isaac Newton. Sir Isaac Newton, the creationist, who wrote twice as much about his faith than he did about science. And he did just an amazing job, and he was the one that coined this word, the photon. The eyes convert photons into the visible spectrum into action potentials. And remember in high school you studied action potentials. It's basically how we send information, for example, from our brain through our nerves to our muscle, telling our muscle to contract. That's all done by action potentials. Photons, right now, striking your retina, giving an image, all of that is due to action potentials. Remember, maybe some of you have had college biology, and I taught this in my classes, what's called the sodium-potassium pump. And the sodium-potassium pump pumps in a 2-to-3 ratio, and it sends a wave of what we call depolarization, a wave of depolarization. And so that's how our bodies are designed by the Lord Jesus to function. Through this action potential, a wave of depolarization in the optic nerve, the wavelength of visible light as we just covered. So now it gets a little bit more detailed here, but we have to get into it if you'll appreciate what has been discovered, and it really is amazing. Quote, the potential changes that initiate action potentials, that is this wave of depolarization in the retina, are generated by the action of light on photosensitive compounds in the cones and the rods. Remember that from high school, the rods and the cones? Boring, but anyway, that's what the rods and the cones are composed of, this photosensitive material, and it's so active metabolically that the rods and the cones are regularly wear out. They wear out just like your car and have to be replaced. So the Creator has done that process, which itself is about a 2-hour presentation, just how this occurs here. But anyway, light on photosensitive compounds in the cones and the rods. When light is absorbed by these substances, their structure changes. And this is why it's so important that somebody in pre-med takes classes like organic chemistry and biochemistry, because when the structure changes, we call that a cis-trans isomerism, gets into very, very complex, right down to the subatomic scale, the molecular scale, and it's quite amazing. Their structure changes, and this triggers a sequence of events that initiates neural activity. Well, folks, this has been going on as you're sitting here, hand over fist, trillions of times per microsecond, just as it's described right there. What you can see right now is what this is all about, but incredibly put in fast forward. So this is one section, it's called the inner nuclear layer, one section of the retina that we're talking about this evening. And the inner nuclear layer, that's what it's called, of the retina, contains the cell bodies of the excitatory and inhibitory. It has to be kind of a give-take process so we don't go nuts. And so the creator has designed these neurons to be either be excited or not to be excited, inhibitory, interneurons, including bipolar cells, horizontal cells, and amacrine cells. And I don't expect you to assimilate all of this, but I'm showing you this just so you can get an appreciation for the complexity, and we're just looking at a small portion of the retina. The amacrine cells that I just mentioned are interneurons, that is in between. So these are interneurons here, they're in between some other neurons, regulate and integrate activity in bipolar and ganglion cells. Now we're getting to a cell that I want you to understand just tonight, very briefly, very basically. These are the ganglion cells right here. These are found, and they have a process coming off called an axon. How many do we have here? 1, 2, 3. So we have 3 axons coming off of these ganglion cells, and it's the axons from these ganglion cells, from your 2 retinas, that make up your optic nerve that's a diameter, as I say, of about a pencil. The ganglion cell layer contains the various types of ganglion cells that are the only output neurons of the retina. Their axons form the optic nerve, as I just mentioned. So even when I was a graduate student, I didn't understand that. But now as they do more and more research, it's not all of the nerve cells in your retina that send out processes and become an optic nerve. Our optic nerves would be about this big. So the Creator, in His infinite wisdom, has produced just the axons from the ganglion cells, right, the base there, that form the axon. Okay, and so here it is, the optic nerve. As you can see, it's pretty thick, coming from the back of the eyeball, going back to the back of the brain. Now there's a section in the back of our brain, back here, that is involved with taking all of this information that you are seeing right now. All that information is being shuttled down through the optic nerve to the back of the brain, where you have 6 layers in the back of the brain that help you to assimilate and understand what you're seeing. And that's a key, because we're going to see that in just a minute. Folks, it's not only what you are seeing through your retina, where this photon, all this light energy photon is striking your retina and causing this biochemical change to occur and sending those action potentials, waves of depolarization back through the optic nerve. That's one part. But then it gets complex. And all of that information has to be processed microsecond by microsecond in the back of the brain. That's why sometimes when people get a trauma to the back of their head, what happens? It affects their vision. Because we have several people like that. And that's what happens, because it's at the back of the brain. Okay, we have to move on. As that infamous King Henry VIII of England told his 6 wives, "I won't keep you long." All right. Sorry. Anything to keep people awake. I kind of throw out these one-liners. But anyway, what we're going to do this evening, very quickly, is look at Mark 8, and we're going to read about the blind man in Mark 8. And what I want to do, because I want this to be all of us to chime in here, is I would like all of us to read this, okay? So I'll begin, and you follow along. Ready? Begin. "And he cometh to Bethsaida, and they bring a blind man unto him, and besought him to touch him. And he took the blind man by the hand and led him out of the town. And when he had spit on his eyes, he put his hands upon him. He asked him if he saw aught. Look at this, verse 24. And he looked up and said, I see men as trees walking. I see men as trees walking. What did he mean? Next verse, read together. After that, he put his hands again upon his eyes and made him look up, and he was restored and saw every man clearly. Folks, this is amazing. Remember what I just finished describing a few minutes ago? That seeing is really a two-step process. The photon energy coming in, striking the retina of our eyes, setting off these depolarization and these biochemicals within the rods and the cones in such a way that information is transmitted through the what nerve? Science is so easy. It's so easy. The optic nerve. Very good. And then that information goes to the front of the brain, the back of the brain, where we have 6 layers of the back of the brain that help to interpret what it is that we're seeing. Isn't it interesting that the Lord Jesus is healing this man's blindness in a two-step process? And we're going to get into that. Jesus asked him, do you see anything? The blind man replied, well, I see men as trees walking. Let's stop right there. He's a blind man, congenital blindness from birth.
How do you know what a tree is?
Well, yeah, we're going to get to that. That's what I'm trying to explain. Yeah, that's it. And so he says, the blind man replied, I see men as trees walking. So this man who's been blind, he knows what a tree is because he probably felt it, right? There's a lot of things that blind people can pick up by tactile stimulation. You're using your fingertips, reading Braille, that sort of thing. And so he had an idea as to the bulk of a tree. And when people would come up and hug him, for example, well, they had a kind of a bulk too, right? I'm just kind of guessing about this. And so this is why he said, I see men as trees walking, because both have this kind of this bulk, as it were. And that's all he could say because he had never seen before. All of a sudden, the Creator, the Lord Jesus Christ, gave him this energy, this light energy through his eyes, and he didn't know really what to think, because when people who have been cured of their blindness, and this is important, we have a lot of people who are cured of their deafness, right? And we watch those really emotional videos on YouTube when a person hears for the first time. Have you seen those? Oh, you're missing out if you haven't seen those. Go to YouTube, not now, and and do that about people who hear for the first time, and I guarantee you'll get out the handkerchief. But it's not that common with people who have lost their sight to get their sight, or if they never had it to begin with. It's not as easy as you think. As a matter of fact, to see after not seeing for a half a century, for example, is so traumatic, is so overwhelming that there are cases of people that wish they hadn't seen at all. You know why? Because it's so overwhelming. See, the first time a person sees, what they see is not, oh, look at this, here are my friends, oh, there's Mildew, my cat, and all this. You know, they don't know, because they don't know what the friends look like. They don't know what a cat or a tree or anything looks like. And so they have been interviewed, and they say, well, everything is just, it's a mishmash of colors that they don't understand because they've never seen colors, and shapes that they've never seen. Can you see how that's traumatic? A person who is given their vision for the first time, you know one of the most traumatic things of them? Stairs. Stairs, because stairs have different shades. And we all understand that easily, we can navigate that, but a person who has been blind and they see those various shades and stairs, they don't know what's going on. They simply don't know what's going on. And so what's happening here is this man is saying, I see men as trees walking, because that's all he could say. He has a very limited amount of information from which to draw. As Keith Manow writes in National Review, This phrase is not a poetic image. Ladies and gentlemen, it is a clinical description. It truly is a clinical description. Like Virgil, and Mano was talking about Virgil, a 15-year-old man who was given sight at the age of 50, having been blind his whole life, and he had had the surgery, and it's very, very rare, as I say. The Bethsaida man could now see, but he cannot yet make sense of what he's seeing. This is why it's so traumatic for people who receive sight for the first time. Your brain is saying, what is going on? The brain is going, I don't understand. Tree and man run together, as did trunk and tree top for Virgil. In short, Mano concludes this is irrefutable evidence that a miracle did occur at Bethsaida. No charlatan in the crowd could have faked it all by pretending to be blind, because only someone recently given his sight would see men as trees walking. A faker, not knowing about the post-blind syndrome, there's a word for that in medicine, would have reported that Jesus gave him perfect vision. Hey, this is great. I recognize all my friends. That simply doesn't work that way. And so Jesus, why did he do it in a two-step manner? I think it's for us in the 20th and 21st century. Because medical technology has progressed in such a way that we now begin to understand the physics and the physiology and, yes, the psychology of vision. So for centuries after this miracle occurred, people, theologians, scientists, nobody really understood what that was all about until now. The hair in the back of your neck goes up. Until now. Like Virgil, that man who's been blind until 50, this blind man could see, but he had the additional complication of the medical condition called agnosia. He could not make sense of what he was seeing. Jesus, having given his eyesight, then heals his agnosia, that is, helping to interpret all that information that is going to the back of his brain where the visual centers are. In one miraculous instant, his brain was taught what the rest of us have learned since childhood. There's a scientific word for this. Wow. Wow. And so this is some of the more exciting things as a zoologist, when I read about that and have an appreciation for what the Lord Jesus has done in the field of zoology, studying physiology and anatomy and all that. This is just, this is amazing. And then the Creation.com, the author in Creation.com said, when sight is suddenly restored, there is the need for the development of some new pathways in the visual cortex of the brain. And so you have the visual cortex of the brain, which has never been used for somebody who's been blind since birth. And now all of a sudden, the visual cortex has to get busy and start to interpret what is being said, which includes development of new neural pathways to the brain. It gets awfully complex, incredibly complex. So how is the brain wired? That's a good question. Understanding how the brain is wired is among the most crucial steps in understanding how the brain encodes information. And it is really something. I do a lot of writing for ICR, and one of my favorite insects, besides, of course, the bombardier beetle, every creationist, yeah, we all love the bombardier beetle. But you know, my favorite insect, a lot of insects carry the immature stages of parasites, and so I love all those too, like the mosquito. But anyway, I don't want to get down that rabbit trail. One of my favorite creatures is the honeybee. The honeybee. One evolutionist said about 5 years ago, he says, these are very, very, he used the word 3 times, very smart creatures. Very smart creatures. How many neurons, how many nerve cells? We have billions, I guess, of neurons in our 3-pound brain that holds our ears apart. How many neurons does a bee have, a honeybee? Well, the honeybee has a brain about the size of a small grass seed. Well, Frank, that isn't telling me much. Okay, 950,000 nerve cells is what a honeybee has. And with those 950,000 nerve cells, bees can recognize human faces. A bee can recognize human faces? Yep. How about counting? Yeah, bees can count. Not to infinity, but they know the difference between 1 and 0, which is more than I ever thought of when you look at a bee. Bees also have that remarkable ability to give information to their fellow bees at the beehive when it comes back after finding a big field of flowers that's full of nectar. How does the bee tell his friends there at the hive? Well, of course, we all saw that in the Moody Bible Institute thing about the dance of the honeybee, and that's never going to go out of date, by the way. But bees do that dance. I never realized this, but they do it in the dark, right? They don't have little lights that they turn on. Okay, gather around. I'm going to show you, it doesn't have that. There's no light there. And so by tactile information, the other bees go up against the bee to kind of feel how it's doing its waggle dance, which way he's going, which way he's turning, and all that, giving out the information. I guess it works even in the dark. Why? We still have honeybees. So it must work. And so bees have this remarkable ability of navigation to be able to recognize human faces, to count. There's a number of other, by the way, they've done some research and found out, hey, bees like to play soccer. Frank, now you're just preaching. It's true, bees like to play soccer. And it's not that they fool around with those little tiny soccer balls because they get some sugar water or something like that. They do it because they like to do it. Well, how in the world do you know that, Frank? Well, what researchers have done is taken some bees, and then they have a long walkway to some sugar water. And on either side of that walkway are soccer fields, bee-sized soccer fields with the balls. And the bees inevitably, instead of going right to the sugar, will go and start playing with the balls and putting them into a goal. Now, you think I'm just preaching, but go to that ultimate source of all authority, YouTube, and type in bee soccer. Bees play soccer. And you'll find out I'm telling the truth. It's amazing. There's a lot more to it than that. But the point is, and I know I'm getting off on a rabbit trail here, when we look at this wiring here, how is it bees have this incredible intellect with only 950,000, plus or minus 2 or 3, nerve cells? And the answer is, as far as scientists can tell, it's how these nerve cells are wired together. It is very, very sophisticated. And through this wiring of these nerve cells, it gives the maximum, the optimum function of these 950,000 nerve cells. It's all within the wiring. And what we then have to ask the question, folks, is that wiring due to time and chance and natural processes, or is that wiring due to plan and purpose and special creation? Those are the only two choices. Long time periods, natural processes, and blind chance? I think not. I think we're looking at plan and purpose and special creation. So this is the wiring diagram of the section of the mammalian brain, and there's nothing simple about this, and this is painfully simple right here. This is incredibly simple. It's a lot more sophisticated than what you see there, but you can get appreciation for what it means. A connectome is the comprehensive map of neural connections in the brain, or if you will, a wiring diagram. And this is more of what it looks like. It's called the connectome. Last year, researchers from the University of Sydney, the University of Queensland, and the University of Cambridge, quote, have confirmed that human brains are naturally wired to perform advanced calculations, much like high-powered computer, to make sense of the world through a process known as Bayesian inference. Bayesian inference. Now we're getting complex. Sorry, but this is just what it's all about here when we start understanding what's happening at the neurological level. And this is written by an individual who's no creationist, and he's saying, yeah, we're naturally wired to perform advanced calculations. How do you get that natural wiring? Time, chance, and natural processes, or plan, purpose, and special creation. I just really don't see, as a zoologist, how you can get this kind of advanced calculations by a cosmic roll of the dice. Bayesian inference, okay? We can't go beyond that until we define what Bayesian inference is. It's a statistical method that combines prior knowledge with updated evidence to make an intelligent guess regarding a hypothesis. All right, put another way, it's a method to assess how new information influences the chances that a current belief or hypothesis continues to be correct. And so a person who has never seen an airplane before, never seen one, but they've read about the dimensions of an airplane, and that's all they've read, and then one day they see what? They see an airplane. Bayesian inference says they are making the inference that that's probably an airplane, based on what I do know. And so that's kind of the idea. This is the mathematical formula for determining what they call conditional probability, and don't worry, we won't get into that. This is one of my favorite slides right here. Because again, as an undergraduate student in biology and then a graduate student in invertebrate zoology, this is incredible. The human brain may be even a more powerful computer than thought. It's even more powerful. Here's what they said, Charles Choi in 2019. Microscopic branches, right here, these microscopic branches of brain cells that were once thought to be basically serve as mere wiring may actually behave as mini computers. The wiring itself behaves like mini computers. Folks, I can just see the naturalistic evolution theory disappearing into the sunset because it simply cannot explain something like this. You know, Paul said in Romans chapter 1 and verse 20 that God's creation is clearly seen, right? Now, Paul could have said, well, God's creation is seen, but Paul took that extra step and he said that God's creation is clearly seen. It's clearly seen. The Sherwin translation of clearly seen is painfully obvious. Okay. And so here we find, you know, the Apostle Paul didn't know about this. You know, they didn't know anything about that in those days, and yet Paul said God's creation is clearly seen. We in the 21st century have even less of an excuse than what natural man and woman had back 2,000 years ago. With what we're seeing here, not to mention, well, I have to mention it, James Webb Space Telescope. Who would be at the forefront, chafing the bit, going, yeah, when the James Webb Space Telescope was launched on Christmas Day, and it went out to orbit 1 million miles away from Earth and set up an orbit out there and started taking knock your socks off pictures that caused all these cosmological textbooks to go out of date with just the first dozen or so pictures. And you remember I was talking about the the infrared range? Well, the infrared range is where the James Webb Space Telescope shines, no pun intended, because all of the basic mechanisms that are in the James Webb Space Telescope are really tuned to the infrared. And we're seeing pictures that nobody ever would have guessed at. But what does the psalmist say in Psalm 19:1? The heavens declare the glory of God. And I'm going, wow. By the way, I just made the reference. I was on a website that talked about James Webb Telescope pictures and was secular, and I just put Psalm 19:1 in the comments, and you should have seen the reaction. I didn't even have the verse written out. People were going nuts, and you saw those laughing faces and all of this. Just Psalm 19:1, and finally, what do they call the person who kind of oversees that? Moderator. The moderator said it all. You know, he got in on it and said, and he banned me. banned me for just saying Psalm 19:1. I got banned. Yeah, that's it. Boy. All right. Well, anyway, this picture shows a computer simulated image of what's called pyramidal neurons in the cerebral cortex. That's a first about half-inch of the topmost portion of the two hemispheres of the brain. Cerebral cortex revealing branching dendrites now shown to carry out sophisticated computations rather than just acting as passive wiring. What's that scientific word again? Wow. Wow. So that was my introduction. Now, let's talk about the brain's amazing ability of visual perception. Had to go through that introduction first. Okay, fasten your seatbelts because we're going to get into some science now. What is visual perception? Well, in psychology, visual perception refers to the brain's ability to interpret and make sense of visual information received from our eyes. It involves recognizing shapes, colors, depth, and interpreting spatial relationships between objects. Did the blind man in Mark chapter 8 have any idea to this? No. And that's why he said, I see men as trees walking. How did he receive all of the rest of the information to interpret it? Well, that's where the Lord Jesus did part 2 of his miracle, so that that blind man in Mark chapter 8 could recognize shapes, colors, depth, and interpret spatial relationships between objects, all within a fraction of a second. Visual perception is crucial for understanding our surroundings and interacting with the environment effectively. So, put another way, a little bit simpler, visual perception is the ability to interpret the surrounding environment through daytime, or what they call in science phototopic vision, color vision, night or scotopic vision, using light in the visible spectrum, that's what we saw at the beginning of the presentation, reflected by objects in the environment. The only reason why you see objects in the environment is that they reflect light, okay? So you remember the visual spectrum, right? And we look outside here, and what color do you see outside? See how easy science is? Green, okay? That is the visual spectrum of light of the chlorophyll molecule that is not used. And so since the green is not used, it's being reflected back to our eyes and we see green. Because of all the visible spectrum of light, during photosynthesis, making life energy from light energy, the chlorophyll molecule is designed by the Lord Jesus, the Creator, not to use green. And so it's reflected back to our eyes. See, so when something shines, when light shines on a surface, one of three things happens. Either the light goes straight through the object, the light is absorbed and used by the object, or light is reflected back from the object. Those are the three choices that happens. So we look at how light is striking the plants out there, and we understand that's what's not being used. It's kind of interesting. And so, yeah, these are some of the pictures. We've all understood this, right? Here's the two ladies, the old lady and the young lady and all that. And okay, just the way that we have this visual perception. This comes from Nature Communications, Volume 14. And Nature Communications is no friend of the Midwest Creation Fellowship. But here's what they said. The human visual system is tasked with inferring environmental attributes from image data that can be corrupted by noise from both internal and external sources. So right now, your vision is we're choosing what we need to see, not a lot of other things that we don't need to see, basically is what it says. To combat the influence of such noise, a statistically optimal or ideal observer would compute the distribution of possible environmental states and combine this prior information with incoming sensory signals to infer the true state of a sense. Okay, so sorry about this, but this is what this article is all about. It's a fascinating article, and it's, well, it's fascinating. But anyway, While this general framework for understanding sensory encoding is relatively uncontroversial, basically scientists understand about the general framework, the biological instantation, that is the expression of an abstraction by a concrete example, and I won't go into it any more than that, of the prior for the environmental statistics is largely mysterious. In other words, there's still, basically what they're saying here, there's still a whole lot we don't know or understand about vision. That's what they're saying, okay? Cutting to the chase. Okay, one or two more like this and then we're almost done. The research team led by William Harrison made the discovery by recording, and the discovery here is this. This is the discovery that they're talking about. The basic structure and connections within our brain's visual system are set up in a way that allows it to perform Bayesian inference, there it is, that phrase again, on the sensory data it receives, okay? That's what they mean when they say this discovery. By recording brain activity from volunteers while they passively viewed displays, engineered to elicit specific neural signals related to visual processing. They then devised mathematical models to compare a spectrum of competing hypotheses about how the human brain perceives vision. So this is what they're doing right now. And it includes just a lot of very detailed biochemistry, a lot of detailed microanatomy and all that. It gives you a renewed appreciation for what the Lord Jesus did in Mark chapter 8. Using data-driven approach, here we extract the brain's representation of visual orientation and compare this with simulations from different sensory coding schemes. Okay, we found that the tuning of the human visual system is highly conditional on stimulus-specific variations in a way that is not predicted by previous proposals. Translated, this means we really are stepping into an area that we don't know much about. That's what they're saying, because they are getting, seeing information in a way that's not predicted by previous proposals. And so all of this shouts what? Design. Just as we do not get blueprints from an explosion in a printing factory. And so here what they're seeing is they're saying our brains have an inherent what? Design. Oops, the D word. Design means what? A designer. Design means a designer. And so they kind of made a mistake here, because they said has an inherent design that allows this advanced form of processing. Creation means a creator. Design means a designer. And so I had to put the blueprints up there because, after all, they said an inherent design. So how did this visual system come about? Okay, we're at the home stretch now. How did this visual system come about? How is it that we can take this photon energy that you see here, comes through the eye, there's the optic nerve, there's a section of the brain that processes this information. Did all of this, folks, come about by time and chance and a cosmic roll of the dice, or was it through plan and purpose and special creation? It sounds so simple, an evolutionist said. Take some light, that is photon energy, and turn it into an understanding of the world around you. We do it every day. I say we do it every microsecond of every day. Yet there isn't a single computer on earth, no matter how powerful, that can mimic the feat of vision to any real extent. So you can have a whole battery of supercomputers and they would not even begin to be able to process the information we take in our eyes in 2 seconds. 2 seconds. The first exascale is defined as that which can perform 1 quintillion operations per second. 1 quintillion. I think that's what was it? The national debt. Okay. 1 quintillion is the number of people who have seen Elvis at Kmart. The first exascale, that is performing 1 quintillion operations per second, computer in Europe is called Jupiter. And it should be completed in this last year. And it may even become the most powerful computer in the world. It's called Jupiter. And even Jupiter is not able to process the information we take in our eyes in less than a second. I said 2 seconds, really it's less than a second. Question of the evening: how does the human brain process visual information right now? Answer: it does so by what we call neural pathways. We can understand that. There's all sorts of neural pathways in the body, and in the brain especially. The axons of retinal ganglion cells, and those are the cells that we talked about briefly, that send all their processes down that makes up the optic nerve, project a detailed spatial representation of the retina on the lateral geniculate body. The lateral geniculate body are those sections on the back of the brain there. Lateral geniculate body. And that's, if those get damaged, that's what causes problems with vision. Each geniculate body contains 6 well-defined layers. Well, I have to include this because this was a zoology text I used. I think it was a first or second edition back when I was in college in 19... and now it's up to its 18th edition, and it's written by not one, but 5 evolutionists. 5 evolutionists. So here's what 5 evolutionists had to say when they talk about homologous genes that underpin eyes, okay? Homologous kind of refers to evolution. There's a lot of serious problems with homology when it comes to evolutionary theory. But they try to explain these homologous genes that underpin the eyes. In other words, why we see. And it gets down to the genetic level. It gets down to these genes, sections of DNA that code for a polypeptide or a protein. And that's horribly simplistic here in the 21st century now that we're learning so much about the nature of the gene, but that's a different presentation. Here's their quote, 5 evolutionists. Listen carefully. Once these 2 genes originated, stop right there, please. Stop. What do you mean, once they originated? Help me in my unbelief, okay? I need to know, as a zoologist, how did we get these 2 genes to originate? That's a serious and a good scientific question. How did they come about? Then they said natural selection, whatever that is, natural selection eventually produced the specialized organs. Okay, wait a minute. Natural selection eventually produced the specialized organs? There's a whole lot of what we call hand waving. It's really what it is. It's hand waving. It's not a scientific explanation. Organs of vertebrates, mollusks, and arthropods. Sounds like a law firm. But anyway, they said, we can have fun tonight. Can we have fun tonight? Specialized organs of vertebrates, mollusks, and arthropods. So really, this is not an explanation, folks. Now, the purpose of ICR, please understand, the purpose of ICR is not to criticize or condemn or make fun of or anything else. But when statements like this are made, they have to be called out, okay? I'm not trying to be sarcastic. That's not our purpose. But when you read something like that, you just have to ask some questions. That's what I'm saying. And so what I would like to say in answer to this statement up here is, can we say that Jesus created the specialized organs of vertebrates, mollusks, and arthropods? That's just as scientific as that, or if you will, this is just as religious as this. We can say it both ways. Both ways, okay? And so we would say, no, I don't, when a creationist and evolutionist are standing side by side and they're looking at a rose, and the evolutionist says, no, the creationist says, wow, look at that rose. Look what God has done. And the evolutionist goes, what are you talking about? Look what natural selection has done. Okay, so natural selection really is the place of God. So they always refer back to natural selection. That's okay. It's a free country. They can do that if they want. But that's not scientific when they say natural selection eventually produced. We would say that Jesus did. Well, they talk about convergent evolution, which again is hand waving. Virtually all triploblastic animals are designed with at least two conserved, and the word conserved is, when you read evolutionary literature, and I've read a bunch, when you see the word conserved, that means unevolved. It didn't evolve. It's conserved over and over, like your hemoglobin molecule that's packed in your 33% of your red blood cell is hemoglobin that's designed by the Creator to carry oxygen. Very good. And so that's a conserved molecule that is unevolved, thankfully. And so two conserved unevolved genes, and here they are. One is rhodopsin, that's a visual pigment. It's also called, in biology, we call it visual purple. And the next one is PAX6. Boy, I'm going to do a presentation on PAX6. And it'd take at least an hour and a half to talk about this. Well, 2 hours. And all we're talking about is a PAX6 gene. And back in '96, when I started reading about that, they called it ILIS, the ILIS gene. But then as they did more investigation research, they said, well, let's call it PAX6. But it is ubiquitous in the kingdom of animals. Ubiquitous. Anything with eyes has this PAX6 gene. It really is quite amazing. And it shouts creation. It really does. It's a master control gene for eye morphogenesis. So all I can say is this evening, and I know we're coming out of time here, evolutionists are clueless as to the origin of these two genes, the rhodopsin and especially PAX6, which is so sophisticated, so utterly amazing. Well, speaking of genes, this is Stephen Meyer's book, Darwin's Doubt. I don't know if anybody's read it. It was published in 2013. It is primo. It is wonderful. It's fantastic. Now, Stephen Meyer is not a young earth creationist by any stretch, but he's done his homework. And on chapter 11 of this textbook, the name of chapter 11 is called Assume a Gene. And he answers the questions that evolutionists like to bring up as to, well, here's how genes originated. And genes just kind of copied themselves, as it were. But that's not an explanation. And here's what Steve Meyer said. It is possible that similar genes have been separately designed to meet similar functional needs in different organismal contexts. Viewed this way, similarity of sequence does not necessarily reflect descent with modification from a common ancestor. Class, what is descent with modification from a common ancestor? What is that? One word. That's evolution. Very good. That's evolution. And so Meyer is saying, no, it doesn't necessarily reflect evolution, but could reflect design in accord with common functional considerations. Common functional considerations. I think of, for example, 20, 30 years ago, evolutionists were just absolutely riding the hobby horse of people in chimpanzees having 98.6% the same DNA. We heard that over and over again, but at least 15 years ago, we shown not to be the case, that we have maybe about 80% of our DNA is the same as chimpanzees. And some people go, 80%, that's kind of high. No, it isn't, because a blue whale has about 80% the same DNA as we have. And rodents have about the same. And any other mammal you can think of, a platypus and all these other creatures too. Well, why is it? Why would we all have the same, at that 80%? Well, the reason is because all those creatures I just mentioned, do they breathe the same air we breathe? Shake your head like this. Yeah. Do they drink the same water we drink? Yeah. How about eating the same food that we eat? Yeah. So if they drink the same water, breathe the same air, eat the same food, then a lot of their DNA has to be exactly the same. When I taught anatomy and physiology at Pensacola Christian College, we got into something called respiratory physiology. Respiratory physiology, how the air that we breathe in, the oxygen gets pushed into, through the lungs into the red blood cell and connects with the hemoglobin molecule. Nothing simple about it. A lot of biochemistry there. Well, the same thing goes on in all those other animals. So they'd have to have the same genes, the same DNA code would have to all be the same. It doesn't mean a common ancestor, it means a common designer. a common designer. So please erase this idea of having 98.6% the same DNA as chimpanzees. That is simply, what do they call it, fake news? It's an urban legend. An urban legend is like drinking 8 glasses of water a day. Don't drink 8 glasses of water a day. That's an urban legend, okay? Don't get me started. Anyway, how do we see all colors? God created people and vertebrates with 3 types of cones, each containing photopigments that are sensitive or tuned to different wavelengths of visible light, such as the short, which is a blue spectrum, medium, the green spectrum, and the long, the red spectrum, okay? And so if we can plot it out on a graph, this is what it would look like. There's the short, the medium, and the long. Kind of reminds me of a midget who was stealing people blind because he was pretending to read their palm reader, and he was a midget, and he was reading their palms and just ripping them off. Police caught him, put him in jail, but he got out because he was a midget. He was able to escape, and so the police said that be on the lookout for a small, medium, at large. Thank you. I'll be here all night. So we have the small, the short, and the medium, and the long spectrum as we see there. Okay. This is where it gets interesting. Right here, these 3 groups. We have people here. I'll say people here. What are these? Bees. These are the bees I was talking about. Bees are just absolutely fascinating. And hummingbirds. Well, I have an hour presentation just on hummingbirds. And by the way, hummingbirds have always been very good. Science is so easy. Hummingbirds have always been hummingbirds. Have they found hummingbird fossils? Yeah. Found lots of hummingbird fossils. What are they? Hummingbird fossils. So hummingbirds have always been hummingbirds. And so they're hummingbirds because they don't know the words. But anyway, these animals have specialized cone cells in their retina that can detect lower frequencies around 300 nanometer wavelength that is in the ultraviolet. So do we see in the ultraviolet? No, we don't. Okay, this is people here. Do bees? Look at that. There we go. 340 nanometers. They can see in the ultraviolet, and so can the hummingbirds. And so this kind of gives you an idea here that have the bees, and they have ommatidia. They don't have rod and cone cells like we do, but they have ommatidia, which are just as complex. Birds have 4 color cone types in their eyes, 4 of them. How many do people have? 3. In theory, this enables birds to disseminate a broad range of colors, including many non-spectral colors. Frank, what's a non-spectral color? Well, it's a color whose hue is not produced by a single wavelength in the visible spectrum, but is instead produced by mixing the longest, that is the red that we looked at, and the shortest, the blue wavelengths. Those are the non-spectral colors. Non-spectral colors are perceived when non-adjacent cone types, sensitive to widely separated parts of the light spectrum, are predominantly stimulated. For people, purple, stimulation of blue and red sensitive cones, is a non-spectral color. But the bird's 4th color cone type that people don't have creates many more possibilities. What does all this mean? It means that birds can see colors that we can't see. Birds have an incredible vista of colors, hues, and shades that we can't even imagine. You can't imagine it. But birds can see in an amazing vista of colors. And it's just incredible. Ken, how do we see yellow? Well, when light strikes an object, it's reflected and refracted. The word refracted means the light ray changes direction when it enters at an angle, and the cones in our eyes respond to the different wavelengths of light. For example, in the daytime, a lemon's light activates both the red and the green cones. What happens when you combine red and green? What color do you get? Any artists here? You get yellow. Okay, you get yellow. Here it is. There's the red, there's the green. What's that? The Young-Helmholtz theory of color vision is based on the existence of three kinds of cones, each containing a different photopigment that is maximally sensitive to one of the three primary colors. The sensation of any given color, for example, yellow, is determined by the relative frequency of the impulses from each of the cone systems. Now, as far as a code for how we see these various colors, we still don't know. That's still a black box, I guess you would say. But this is called the trichromatic color theory. So colors have three attributes: hue, intensity, and saturation. For any color, there is a complementary color that, when properly mixed with it, produces a sensation of white. And so this is the conclusion from an article written by an evolutionist by the name of Ritchie at the University of Sydney. And this is what this whole second half of my presentation is all about, is to conclude what they were saying, because they're evolutionists. There was no evolutionary description, detailed or otherwise, in this article as to how the human brain processes visual information. No evolutionary explanation. This causes one to ask why the title of the University of Sydney article is "Evolution Wired Human Brains to Act Like Supercomputers." Well, folks, if they never have an explanation within the body of the article describing step by step an evolutionary process that resulted in this, then why would they put this kind of title on their "Evolution Wired Human Brains to Act Like Supercomputers"? That's almost being intellectually dishonest when you go to the body of the article and find that there's no evolutionary explanation. So we would say the correct title would be "Jesus Wired Human Brains to Act Like Supercomputers." Okay, that's what we would say. Now, folks, I know I'm a little bit out of time here, but let me share with you some of the books that we have out in the book table there. And this is a DVD, and we only have one or two more of these left. Dr. Tim Clary has put together his theory of megasequences. And he is the only one really to put down the megatheory sequence in a way after he's interpreted the drilling that has been going on in the sedimentary rock units throughout the world. And it's taken Tim about 2 years to do that. This is a great DVD, and I urge you to get that. Also, what we have available is Creation Basics and Beyond. This is for those who want to take an extra step into creation science and read about creation science in detail. There's no pictures or anything. There's just a series of articles, Creation Basics and Beyond, an in-depth look at science, origins, as compared to evolutionary theory. So this makes for a great read for those who are really interested in creation science. Then we also have The Human Body, and this is a book that is really neat for your kids, your grandkids, your great-grandkids, or your pretty good grandkids. Thank you. Yeah, and so a good color series of a book that will help you help the kids and you to understand the human body and how it's designed. Some guy named Sherwin wrote a book on the oceans. It's been revised, updated, and expanded. And so when I wrote this, we were in San Diego, and so I had the whole ocean at my side there. And it was one of the more fun books that I wrote for ICR, the ocean book. And so feel free to avail yourself on that. Then I also wrote a book on animals, since I'm a zoologist, animals, and children will enjoy reading that. You can read that with your kids and grandkids and all. Then the late Dr. John Morris and I wrote a book on fossils. I love fossils, and I was honored and privileged to be able to co-author this book on fossils with Dr. John. And this is a coffee table book. This has some beautiful, beautiful color plates. And at the end, I wrote about the last 1/3 of the book, where I answered the so-called missing links. Evolutionists say this is a missing link and this fossil is a missing link. And so for a year, I went through the secular literature, I didn't go to creation literature, and showed why these were not missing links at all. And so if you're interested in that, the last 1/3 of this book, I answer the more popular missing link arguments and use the citation from secular authors there. So The fossil record. Who here remembers those plastic platters that you spin on a turntable? What do they call those? Records, yeah, record player, that's right. Well, I used to work at a radio station in Gunnison, Colorado. 827 is our time. And so I would play records and all that. People would call me up and ask me to play the fossil record. But I said, no, we don't play rock music. Thank you. So anyway, okay, now this is for guys. Ladies, you can just shut down, because this is just for the guys. Guys, I know that your wife has probably told you, when we come to these meetings, I don't want you buying all these books. You can get one book. The reason why I say that is that's exactly what my wife says. And as a good husband, I salute, no. But okay, guys, so if you're only allowed to get one book, I can understand that. If one book, then these are the 4 books I would get. Animals, ocean, the fossil record, and the human body. You've got to get them all, okay? And build up that creation science library. Kenny, I'm turning it over to you.